Okay, so I will talk on this topic, physicochemical and biological functionalities of bee products. Food as medicine. It's very interesting this because prevention is better than cure. And here we see some synergistic effects with medicinal plants, species, food plants, aromatic plants, aromatherapy, chemotherapy, phytotherapy, and some products, api products. So it is with our Mediterranean diet, so it will be a very good cocktail to make prevention more than cure. And this synergistic impact will be beneficial for our population. So for modern apitherapy, all beehive products have a medicinal use. Honey, pollen, propolis, royal jelly, bees, apilarnine, queen larva, apiaire, all good. But the problem is how to use these products for nutrition, health, quality of life, prevention, and treatment of different pathologies. So we have quality, concentration, dose as function of time, and we have synergy. But I will talk today only for this relationship between biomolecules and biological activities. As you know, pathologies promote overproduction of free radicals, reactive oxygen species. And if the oxidants are more than the antioxidants, the body faces oxidative stress. And we have enzymatic compounds and non-enzymatic compounds. And in our body, we have superoxide dismutase glutation, catalase, peroxidase as enzymatic antioxidants. And we have another non-enzymatic like vitamin E, C, E, flavonoid, and other polyphenols, which derived from our food, from our apitherapy products. So it will be very interesting to have antioxidant to face to these oxidative stress pathologies. With honey, we have minerals in honey, calcium, iron, magnesium, zinc, because with COVID-19, we have zinc as target for management. And we have polyphenols, flavonoids, quercetin, rutin, galantine. We have also phenolic acids and phenolic acid derivatives like syringic acid, vanillic acid. But the ORAC activity as marker of antioxidant activity from Moroccan honey, we have salvia, Nigella sativa, oreganum, carob, euphorbia, very good, ziziphus lotus, peganum harmarum. So very interesting honey. But the problem with this, like I told before, the main components defined if identified in honey, we have minerals like zinc, selenium, and with this we have marker electrical conductivity and ash content. We have also the compounds of glucose, fructose, glucoside, and this is very good also for marker with HMF. We have organic acid, which is the marker of pH and acidity, amino acid and proteins, and we have apalbumins as immunomodulators from honey and royal jelly. And we have vitamins and polyphenols, which are markers for bioactivity. So we should have this quality of polyphenol compounds in order to make relationship with bioactivities. In our first paper with the serotonic acidica honey, it's carob honey, we have collected all samples from Morocco, and we have also related with the climate change, and we have measured physicochemical properties, mineral content, antioxidant activity in order to select the best honey to be investigated for biological activities. And here we have the protective effects of Morocco carob honey against lead-induced anemia and hepatotoxicity. And here, as example, we have hemoglobin. When we make toxicity with lead, and when we give honey with lead, we make restoration of hemoglobin like in control rates after 24 days of treatment. But it is only aqueous extract. Here also we have aqueous extract in CCL for induced kidney and liver injury. And here it is very important, as I told in the introduction, we have CCL4 rates, and we have honey with CCL4. And here we have in kidney and liver, we have a decrease in antioxidant enzyme like catalase, like GSH, and like GPX. And the effects are very important in liver more than in kidney. But when we treat with honey in hepatotoxicity rates, we have an increase in antioxidants like catalase, GPX, and GSH. And the increase is very important at the level of kidney. So this balance with antioxidants and also with minerals in carob honey gives hope for treating detoxification, diuresis, and cardiovascular pathologies. But here we have ethyl acetate extract, which is very rich in polyphenols. And when we use this in streptozotocin rates, diabetic rates, we have a very good decrease in glycemia. And in control, we don't have any change, but in diabetic rates, we have a very interesting decrease, like in control rates. This may pave the way to investigate therapeutic effects on this extract in diabetic animals and also in patients with diabetes, liver disease, and lipid disorder. Now I will give you, in apitherapy session in Morocco, I have speak on Zendaz honey. Zendaz honey is produced in our region, and we make a very, very good research with this Zendaz honey. It is from Apiaceae family. And this Zendaz honey have a very good physicochemical characteristics and correlation with anti-proliferative and antioxidant activity. And this year, we have this publication in Food Chemistry, very high impact factor. And here we have pollen analysis to have these strategies to make pollen analysis and physicochemical characteristics of Zendaz honey samples. We have pollen analysis like we have majority with Buplorum spinosum. We have Cytisus, Populus. Physicochemical characteristic, we have minimum and maximum because here we have more than 40 samples. We have pH, free acidity, minerals, we have magnesium, zinc, sugar, fructose, millibiose, and arabinose, all this. But the characteristics of this honey, it is very rich in methylserangate. So here we have 4 samples which are rich in methylserangate. And when you look at phenolic composition, we have methylserangate, it's rich in manuka honey and in asphodel honey. And this is very important for bioactivity of this honey. We have quercetin, we have conferol, genistein, we have also catechin, serangic acid, epicatechin acid, but the amount of polyphenol, more than 54% are with methylserangate. So when you use in vitro assay with MMT assay to determine antiproliferative effects in THP cells, which derive from cancer leukemia, and we make incubation after 24 hours and 72 hours, we have a very good decrease in proliferative cells. We have a very good decrease with cellular viability. And when you look at the level of contrast microscope, we show very decrease in viability cells as proportionality with concentration of honey. This is very interesting because we have this proliferation, it's decreased. We have H1 honey, which is very rich in methylserangate acid. We have H2 and H4 honey, which exhibit very good antiproliferative effects with respect to concentration. But we have the EC50, it's the concentration with inhibit 50% proliferative effect. We have the best is with H1 and with H4 and with H6. It's the best honey which give this anti-proliferative effect. But when we look at the antioxidant activity of Zenda's honey in cancer, breast cancer cells, expressed as cellular antioxidant activity, quercetin equivalent by milligram gram honey, here we make at the level of cells, we give oxidative stress in order to have increase in free radicals. And after we measure by fluorescence the quantity of antioxidant activity at the level of the cell. It's a very good marker. As you see, we have H1, H2, and H6 have a very high antioxidant cellular activity, but H3, 4, and 5, it's the same to decrease. So it is very important to have this component in order to make correlation between bioactivity and proliferative. Here we have principal component analysis using the phenolic composition and biological activities of Zenda's honey samples. It is very important to see here, we have in PC1 compound, more than half of compounds are correlated negatively, which form a group with gallic acid, methylxyrangate, cachesin, gallic acid, in addition to color and melanoid. This cluster shows a positive correlation with the three variables as anti-proliferative activity, 1 sur IC50, antioxidant activity at the level of cell, and MTT interference. So it is the good group. And we've shown here that H1 honey is the best. The PCA also show that honey was the best one with respect to those biological activities, and high content on methylxyrangate, gallic acid, while on the opposite side of the graph was H5, which was biological activity and low amount of methylxyrangate and gallic acid. So this PCA gives us a very good correlation between polyphenols, between colors, because with this honey is very dark, so colors and melanoids are positively correlated with biological activity. The Zenda's honey is a traditional local product that have interesting health promoting properties such as antioxidants, anti-proliferative activities. This honey is characterized by its very rich content in methylxyrangate, a polyphenol that has been previously found in Manuka honey, Canuka honey, and Asphodel honey. This multivariable analysis showed positive correlation between methylxyrangate and biological activity. The characterization of this honey as a health-promoting food due to its anti-proliferative and antioxidant activities should increase its commercial value, which would have a very positive economic impact on the rural communities of Morocco, where is it produced. Now I give briefly another example of aro honey, aro miel. We have synergy with aromatic plants and honey. Very interesting, because we should have biomolecules from honey different from biomolecules from essential oils in order to have this synergy. So we have honey from Euphorbia honey from Morocco. We have make HPLC analysis. We find syringic acid, ferulic acid, pyragol, and in essential oils of oreganum, we have the compound of carvacol and thymol. And this notion is a synergistic mixture of honey, 1%, 1 gram by 100 gram of honey. And this mixture, it's very interesting, but when we make combination, we have made in 100 combination, only 9% gives a synergy. So it is a very good article with honey antibacterial effect boosting oreganum vulgar essential oils. And here, it's in French, I will translate. We have the PCA analysis. We have honey with tannic, ferulic, and gallic acid epicatechin are very rich in this compound, make a very good synergistic effect with essential oils to treat Staphylococcus aureus, and honey with pyragol, with caffeic acid, coumaric acid, and rosmarinic acid are very potent to cure pathologies against Escherichia coli. So it is a good study to make this correlation with all parameters. And here we have just this publication, on January 21. We have this combination in CCL4 induced toxicity, liver, and also kidney toxicity, as I mentioned with carob honey. And here we have a very good result that synergistic effect with essential oils and honey give a very good restoration of potassium, sodium, HDL, cholesterol, and we have a very good effect all in the liver enzyme. And when you look at this protective effect of honey against this toxicity with liver markers, I give only this example. We show here CCL4, and we have a very good increase in red, a very good increase in hepatic enzymes. But when we treat with honey, we have a decrease. But when we treat with aromiel, we have a very good decrease in CCL4 treated rate. Also for the three enzymes. So it is very good effect and synergistic effect with honey. And the mechanism of action, we have in our honey, we found ferulic acid, epicatechin, galate, and in essential oils, we find thymol and carbachol. All these are antioxidants with lipid peroxidation dependent pathway at the level of mitochondrial dysfunction. And we have CCL4, which induces lipid peroxidation independent pathway. So we have stress and hepatocyte swelling and induction of stress defense related gene. So it is good because this antioxidant prevents lipid peroxidation induced by CCL4. And here we have this mixture, Thymus vulgaris honey, which have antioxidant activity, oregano vulgar essential oils, which have also antioxidant activity. And when we put it, you have the carbachol, thymol, and terpinin as biomolecules from essential oils. And we have epicatechin agalate in the first time detected in Thymus vulgaris honey. And in CCL4 untreated rate, we have impairment of increase in total protein, in improved liver function, and decrease cholesterol. But when we treat with CCL4, we have restoration of this dysfunction at the level of kidney and liver. So it is very good combination, but we should have the mechanism of action. Here, rapidly, I give with bee bread as functional food. Bee bread also have a very important effect on anemia, on weight disorder, menopause, colitic, hepatitis, immune systems. And we have done a very good effect with hepatotoxicity induced by aluminum. And here I give only an example. With bee bread, we have a dose-dependent effect, and we have a very good increase in hematocrit when we give bee bread at the level of 500 milligrams by kilo. But we should have the proximate composition, energetic value, and also micro elements of this bee bread. We have shown this. We have also fatty acid and tocopherol rich compound. We have quercetin as a compound, conferol in the polyphenol compound of bee bread. And here we just this month we have this article of hypoglycemic, hypolipidemic, and hepatoprotective effect of bee bread in diabetic rate. We have a very good effect, more than glibenclamide. Here also we have this publication with fresh bee pollen with renal function. Rapidly I give you this because it is very interesting. We have selected more than 20 monofloral bee pollen, and here we have a very good amount of zinc, potassium, magnesium, and sodium. And this mineral restore the altered plasma electrolyte and maintain them in the physiological level. And when you give diuretic effect, it's very interesting. We have furosemide, it's a medication with its diuretic, but when we give pollen at 100 milligram or 250 milligram, we have a very good increase in diuresis, 20. at day 30, and we have furosemide, hypokalemia with furosemide, but with pollen, we have restoration of this hypokalemia. We have increase in electrolyte secretion and maintain blood potassium at good level. And we have here the antioxidant parameters and polyphenols and flavonoids, and this high antioxidant activity have strong diuretic activity and prevent against kidney oxidative stress relative pathologies. So it is very good with the pollen to have this nephroprotection and to have antioxidant, anti-inflammatory, blood pressure reduction, and increase in nitric oxide. And we have these physicochemical properties of fresh pollen, and we have a high content of flavonoid, mainly quercetin, which affects renal factors, which promotes diuresis and natriuresis, and also prevent hypertension and cardiovascular disease. So in conclusion, chemical characteristics of bioproducts may allow the extract to be used as bioactive ingredient in the food industry, but they also present potential of the pharmaceutical or nutraceutical sectors for the prevention or treatment of health disorder. And thank you for your attention.
Yes, very good, Badia.
Excuse me, I had been long because I would like to give you a novel approach with the synergy with the honey and medicinal plants, aromatic plants. As Professor Ali asked me, I will give you some data of this. It's very important to have
Thank you very much. Synergism is very important issue. We should talk more. I hope you will have time, Professor Badia Lucy. At the end of the day, in the meeting room, I think many people would like to meet with you and discuss.
Inshallah, I will.
In the meeting room. Thank you. Yes, you are the moderator, so you can continue, please.
So now it's my great pleasure to give to Professor Banu, my good friends and my future collaborators, to give the floor for you to give your lecture. Thank you.
Thank you very much, respected chair. I offer my regards to all distinguished participants and thanks to the organizing committee. And now I'm sharing my screen. I hope it will be seen. Yes, today I will explain some details about our research we made in last year, chemical compositions, the toxic, antioxidant, and antiviral effects, propolis samples from different regions of Anatolia. These produce propolis, a sticky wax-like substance, by collecting leaf buds, resin of plants with their own oral enzymes and saliva. The colors and contents of propolis vary upon plant source and area. As a natural consequence, its biological activity also differs from region to region. Over time, as we know, propolis has been used in traditional medicine as an anti-cancer, antioxidant, anti-inflammatory, anesthetic, antiviral, antimutagenic, antibacterial, and cytotoxic drug. And the purpose of present study is comparatively investigate the chemical profiles and biological activities of 39 samples which were collected from different geographical regions of Anatolia from non-migratory beekeepers, and try to create relationship between plant sources and propolis samples. The steps to achieve this goal were as follows. Volatile components of propolis determined flavonoid, phenolic, and triterpene profiles, antioxidant activity, cytotoxicity was against breast, prostate, lung, cervix, and non-tumorigenic cell line, human embryonic kidney cell, the inhibition of the inflammatory mediator nitric oxide. Antiviral activities were also measured as virucidal activity by in ovo against avian coronavirus.
Tamam hocam.
According to the result of LC-HRMS analysis, while the major flavonoids were found as isosaponetin, naringenin, rhamnocitrin, diosmetin, syrizin, methylquercetin, and apacetin, the main phenolic was found as caffeic acid. The major triterpene compounds were also detected as olenoic acid and heterogenin. Volatile components, alpha-pinan, beta-pinan, and limonan were found as the main compounds in samples. Most of the propolis extracts exhibited more than 80% BPPH scavenging activities, almost similar or higher than the positive controls. The highest antioxidant capacity was found in samples in Izmir, Bergama. Many studies presented that Turkish propolis extracts showed high antioxidant activities due to their total phenolic and flavonoid contents. As you can see it here, difference in chemical composition in regions, higher amount of limonan belongs to the terpene groups found in citrus trees was determined at high levels in propolis obtained from Kelkit, Rize, Artvin, and Trabzon. It has been determined in the propolis samples, especially obtained from the regions where citrus trees grown in the eastern Black Sea region are concentrated. The high level of linalol, thymol, and carvacrol detected in the propolis sample taken from Burdur prediction that astragalus plant, lavender field, and juniper and cypress trees, which are widely found in the region and contain these active ingredients, alpha-pinan present in the leaves of beech and poplar trees were also found at a very high level in the propolis sample obtained from Sivas region. Alpha-cedrin is found in the leaves of the cedar tree, Cedrus libani, was found at high levels only in the propolis samples obtained from Kars. In the high and the mountainous areas of the region, it is reported that alpine herbaceous plants are also seen along with plant spaces belonging to steppe, subalpine, and alpine vegetation. Methylquercetin is one of the phytochemicals with antioxidant effects, very rich in polyphenols, found in Cretaegus and Aesculus hyponocastum, horse chestnut plants, was detected at high levels in samples taken from Hakkari, Iğdır, and Izmir. Also, epigallocatechin has been determined only in Izmir, Manisa, Trabzon, and Gümüşhane. Quercetin content was also found to be highest value in the propolis sample obtained from Hakkari. Manisa propolis contains significantly different on fumaric acid compared to other propolis groups. Fumaric acid is an active ingredient in Arabidopsis thaliana and Punica granatum plants. The highest benzaldehyde content was determined in the sample taken from Artvin, and it was predicted that this might be caused by the presence of Prosimum alicastrum and Picea orientalis plants, which are widely found in the region. Heterogenin, which is the active ingredient of Hederahelix, Quercus, Salix, Nigella, Sativa, and Salvia, detected in high levels in propolis samples obtained from Tunceli and Bingöl regions. And from Hakkari region, the propolis contains significantly more syrizin, caffeic acid phenetyl ester, apigenin, apacetin, quercetin, naringenin, rhamnocitrin, and diosmetin than any other sample. The fact that some plants in the region, Juglans regia, Quercus, Ariganum vulgare, Astragalus, Elaeagnus angustifolia, Cotoneaster, and Morus alba, contain these active ingredients is considered as a reflection of the propolis sample. The highest amount of ellagic acid was found in the propolis produced in the Caucasian bee. Situation makes us conclude that especially Sidonia oblonga, Rosa canina, Rubus canaskans, Cotoneaster, Diospyros lotus, plants in that region are benefited as a source of propolis. In the present study, we demonstrated also the cytotoxic effects of Turkish propolis on 4 human cancerous cell lines and 1 non-cancerous cell line. Also, we demonstrated the anti-inflammatory effect with inhibition of NO production in macrophage cells. Significant cytotoxicity against prostate, cervix, lung, breast cells were observed and also exhibited cytotoxicity against non-tumorigenic human embryonic kidney cell, but generally cancerous cells had higher IC50 values. NO production was suppressed effectively by Anatolian propolis samples on LPS activated macrophage cells, and the best result was found in Ankara sample. Many studies have demonstrated that Anatolian propolis has a significant high effect in terms of antibacterial and antifungal activity. In this study, the antiviral effect of different Anatolian propolis was evaluated for the first time against avian coronavirus in OVO. Studies on the antiviral activity of propolis have showed that it caused a decrease in the viral replication herpes virus. This study was important in testing the antiviral effect of propolis samples collected from different regions against chicken coronavirus. According to our virucidal activity results, the best effect was found in Uşak propolis. It could be possible with Caucalis platycarpos plant, which contains very high antiviral activity in this region. When the chemical content of the examined propolis samples compared, it may cause from the flavonoids content or from the especially synergistic effects between the compounds or unknown compounds. These results indicated that the molecular content of propolis is the main factor on avian coronavirus. In the light of data obtained in the study, it is expected that more detailed studies will be carried out in each region according to the seasons and different localities that will provide some critical information on the preparation of the local Anatolian propolis product for different applications, such as medicine, cosmetics, and food industry. In this respect, it is predicted that the potential of Anatolian propolis has been proven with this study and will lead the development of locally registered geo-branded products in the future and will provide significant support to local producers. More research will be needed to see the extensive results about on this topic. Thank you very much for your attention, and I would like to also thank our working team to make this research. Thank you very much.
Yes.
Stop the chat. Okay. Thank you, Professor Banu, for this excellent presentation. It's very interesting because we have volatile compounds and we have other phenolics. So it will be interesting to make extraction of essential oil of propolis because it is very little. We have made this also, and we could put it with other products. Very, very interesting study. Thank you.
Thank you.
So now let's move to Professor Otilia Bobis. The floor is yours.
So I will be as short as I can. Dear Api friend, I'm very glad to be here, and I want to thank Stefan and Professor Timucin for asking me to be part of this wonderful congress until now, what I saw. Today I want to present to you a small part of the work made by the team from Api's laboratory. It's our laboratory from the University of Agricultural Science and Veterinary Medicine from Cluj-Napoca. laboratory for quality control of bee products. Also, I want to mention that I make this with this presentation. I'm make an in memoriam of Professor Liviu Mărghitaș, head of our department and director of laboratory control of bee products until 2016, which passed away recently, and to whom we all, the department, the laboratory, are grateful for all that he wants for the team, and making us the specialists that we all are now. Thank you. Next, please. So, laboratory for quality control of bee products belong to the University of Agriculture Science and Veterinary Medicine from Cluj-Napoca. It's located in a modern institute, Life Science Institute. It is accredited for tests made for beekeepers, for customers, for buyers and processors. Here we derulate some important research projects we conducted here over the time, and also different PhD theses and practical works with students. Thank you. You can move on, I think, 3 times. Okay. Yes. Now, because all here are specialists in bee products and in their chemical composition and their bioactivity determination, we know that the main bee products are honey, propolis, pollen, royal jelly, and wax. Forward, please. And it is known that the nutritive value of food products is given by the content of sugars, lipids, and proteins. With these parameters, the nutritional or energetic value of the product can be calculated. Please go on. And beside the nutritive value of the bee products, we all know that biologically active substances are very, very important. And I think the highest contribution is given by the polyphenols and by natural pigments in bee products. Of course, all others, organic acid, vitamins, enzyme, polyphenols, essential oil, and aroma substances have also an important value. Next, please. For the determination of polyphenols from honey, modern techniques are recently used, like high-performance liquid chromatography with difference detectors. They can detect, this technique can detect very small amounts of these phenolic compounds. For example, using this technique in our lab, we managed to identify the presence of different phenolic acids such as caffeic, paracumaric, ferulic, and also different flavonoids such as pinochembrin, apigenin, kaempferol, chrysine, and all others. Go on, please. Using the same technique for propolis, we can identify the same phenolic acids, but in amount much higher than in honey. So we have here different phenolic acid like caffeic, paracumaric, ferulic, or which is very important, different flavonoids. And we saw here, and in the next day, I hope we will see more, the presence of quercetin, which is very, very important nowadays with this pandemic. We all know what the quercetin is doing in this fighting in this against this virus. And also we can detect the caffeic acid phenethyl ester, which is a common compound for propolis, and it's known to have the anti-carcinogenic effects. So that's why the propolis is so good. Okay. Next, please. If we move on with the composition and benefits of bee products, we reach the bee-collected pollen and also bee bread, possessing very high amounts of vitamin C, beta-carotene, and lutein. I'm sure all of you heard about these pharmaceutical products. In pollen, we have all these in natural form. Also can be detected using this technique a long list of complex of polyphenolic substances, which give the extraordinary properties of fresh bee pollen. Bee bread also has a similar composition, but a higher biodisponibility for organisms due to the fermentation processes that occur in the transformation of bee pollen into bee bread. Here we have a higher amount of free amino acids and free unsaturated fatty acids omega type. Please go on. Regarding the composition of royal jelly. Royal jelly is known especially for the presence of 10-hydroxydecenoic acid and some specific proteins called major royal jelly proteins, which can be affected if the product is not entirely natural, if some adulteration is occurring. In our laboratory, using HPLC and also electrophoresis, these compounds of royal jelly may be determined. So all this determination made in our laboratory as research project, PhD thesis, and regular analysis have as results different publication over the time in different renowned journals of high rank. Please go on. One of further, please. One of our study on honey was published in Journal of Agricultural and Food Chemistry, where different unifloral and multifloral honeys from Romania were evaluating using HPLC their phenolic profile, but also organic acids before and after fermentation into the products, especially in the production of mead and honey vinegar. Conclusion of this study was that different profile of biologically active substances are obtained from different types of honey. So in the production of mead and vinegar, it's very important if we know what we expect to find. It is very good to have a certain raw material, so a certain type of honey. Further, please. Now I want to mention one of the first published studies of our group regarding different honeys from Romania, which was made in food chemistry. Here, physical, chemical, and bioactive properties of different honeys were evaluated for the first time in our laboratory. This was back in 2009. This paper was several years in Web of Science, a paper, highly cited paper for its section, until now gathering almost 400 citation. We are very proud of this article. Please go on. A review article made by our research team with an interesting topic was published in Oxidative Medicine and Cellular Longevity, a journal from Hindavi Publishing Group. Here, different studies were gathered to explain if honey may or not be used by people suffering from diabetes mellitus. And the answer is yes, honey may be used in small amounts by persons with type 2 diabetes, especially acacia honey or other honeys with very high content of fructose, over 40%. So this is very important to know. So diabetic people may consume small amounts of honey, but honeys with high content of fructose, such as acacia honey or citrus. I think citrus honey have also this high amount of fructose. Further, please. As I have mentioned of acacia honey from Romania, this type of honey is known as a very good quality bee product, being awarded in many countries with important awards and medals. In the study that I am presenting here, we have demonstrated the quality of acacia honey from Romania by analyzing, beside the physicochemical composition, also the nutritional value and the volatile profile, which may be used as marker of geographical origin for acacia honey from Romania. This study, as you can see, was published recently in Foods Journal. Please go on. Another review study was published in the renowned Taylor and Francis journal Food Reviews International. This review was made for the demands of honey labeling. It is known that the origin of honey and the production methods are very important features for establishing the economic value, nutritional value, as well as the whole quality issue. Therefore, labels must be informative for consumers, presenting both the botanical origin and geographical origin, and also maybe mention the nutritional value, which came from the chemical composition. I know that in the other section, colleagues from Turkey said that it's very important for propolis products to be correctly labeled, but this can be applied also for honey. So it is very, very important that we have a good labeling for all bee products, in fact. Please go on. Regarding the bee pollen and bee bread, different studies were made by our researchers to evaluate the composition and the bioactivity of pollen and bee bread. Again, in food chemistry, we published one of our first studies on bee pollen from Romania. I want to say that Romania is known for its production of fresh bee pollen, because we have a variety of landforms and a very rich botanical flora. due to the fact that a large area of Romania consists of hills, meadows, and mountains, and the flora is very rich and very diverse. We know that this is representing the heaven for bees. Nowadays, I know that Romania exports high quantities of fresh pollen in Western countries, and not only here. Further, please. Now I want to show you two papers which were made in our laboratory by, well, together with the whole team, but my colleague Rodica Mărgăoan, who is a specialist in palynology. She was formed in laboratory for quality control of bee products, where she had also her PhD thesis. Published in the renowned Journal of Agricultural and Food Chemistry, a study regarding predominant and secondary pollen botanical origin that may influence that may influence the carotenoid profile and also the fatty acid profile for bee collected pollen. So Romanian bee pollen originating from different botanical sources, of course, have these different amounts of fatty acids, omega fatty acids, and also different amounts of carotenoids, which two classes of compounds which are very, very important for both for nutritional but also for biological activity of this bee product. Further, please. And if we talk about the nutritional value, also the antioxidant properties were demonstrated in different studies. And here in this study, the antiproliferative and apoptotic effects of bee pollen extracts on different tumor cell line was investigated and demonstrated. This study was published in a Journal of Apicultural Science. Further, please. Now I want to show you our latest study on bee bread composition. Was published this year in Food Science and Technology from Elsevier. We demonstrate in this study the importance of botanical origin determination and how this may influence the antioxidant activity and generally all the biological activity, together with the chemical characteristics and of course nutritional values for bee bread. Further, please. I'm very proud of this review article published in Evidence-Based Complementary and Alternative Medicine, a renowned journal from Hindawi Publishing House. We made this review containing all what was published until that date about Romanian propolis. So chemical composition, bioactivity, and different medical uses. This is a reference study gathering the research made on poplar type propolis all over Romania. So it's the study was made by our group, but it contains what was published in all research groups, university scientists in Romania at that time. Okay, please go further. We are talking about poplar type propolis, Romanian propolis. In this study, this was evaluated for its phenolic profile, its antimicrobial properties on different bacterial strains from humans, but also different bacteria from bee diseases. In this study, we demonstrate the importance of geographical origin and also the plant resin source, which is very important, on the bioactive properties of propolis extracts. As you can see, this study was published in Journal of Apicultural Research. Besides the, please go further. Besides the chemical composition determination, the bioactivity of all the bee products, our laboratory and research team have also interest in bee health and finding alternative treatments for bee diseases. We know that we have to have healthy bees in order to have safe and high quality bee products. Due to the fact that bees, as any other living soul, may suffer from different diseases, and their treatment must not include antibiotics due to the remnants of these contaminants in the bee products, in honey, in pollen, and bee bread, and even wax. In our research projects, we try to see if we can find alternative treatments using medicinal plants or even products from the hive itself that could be used in bee diseases treatments. It was observed in some studies. We have observed that bay laurel extracts may have antiviral activity. This study was published in a virus research journal, and also using certain honey type or propolis in artificial bee feeding. Those may act like self-medication in different bee disease pathology. In those studies, it was demonstrated that sunflower honey reduced the bacterial load of infested bees, bees infested with the nosema spore, so artificially infested. This type of honey having both in vitro and in vivo antibacterial demonstrated activity. These studies were published in also very good journals, Ecology and Evolution, and Behavior Ecology and Sociobiology. Please go on. As I told you at the beginning, this was just a short presentation of our work, of the laboratory work. We are available for any collaboration, and we had different collaboration with the specialists from different countries. Some of them you could see on the authors of different publications, and we are open also for the future with this collaboration. Please go on. I want to show you a dear picture for me. This was the beginning of AFI's laboratory. We were young, but now we are specialists, we are researchers, we are academics. And again, I want to thank Professor Marghitash, because under his supervision, we are what we are now. Thank you very much for your attention.
Thank you, Professor Otilia, for this nice lecture, because you give all the analytical studies. So it will be a good opportunity to make intercomparison with bee products from our country, and to select and to make a consortium in order to make standardization of bee products, like pollen and bee bread. We have seen the same compounds with the vitamin, tocopherols, fatty acids. So it will be interesting to collaborate for pharmacology and at the level of in vitro studies, inshallah. Thank you.
Thank you also.
Yes, one just word before we go to the next section. The idea presented by Ottilia that the bees can be self-treated with their own bee products. We have apitherapy for humans. We have apitherapy for animals. But look, you have also apitherapy for the bees. So
Yes.
we can use the same principles. And it's very, very nice ideas and very good ones. Okay. Yeah. Badia, I think we can close this section.
Okay.
And because we need to go to the next one.
Okay,
And
so thank you very much for this nice panel and for this instructive communications and publications. It will be very good to make a collaboration with Banu and Ottilia and me. No, why not? To make in the next conference in Fez, I will invite you, and Stefan will be the leader. Stefan will be the leader. Thank you very much. We close the session. Thank you. Thank you.
Okay, thank you.
Bye.
Bye.